Welcome to the Deep Dive, the show where we take a stack of dense academic source material and really just extract the essential knowledge you need to be truly well-informed.
And today, we are definitely doing a deep dive.
We're getting into the foundational world of microscopic anatomy, what we all call histology.
That's our mission today. We're going to demystify the entire workflow, all the methods that turn a raw piece of biological tissue into those stunning, highly informative images that really define modern biology.
Because that's what histology is at its heart, right? The study of microscopic structure.
Exactly. And the goal is always to connect the precise structure you see under the microscope with the function that structure actually performs.
And when we talk about the methods to do that, I mean, the landscape today has just exploded. The sources we have are fantastic because they don't just stick to the old school light microscope.
Not at all. They highlight this incredible diversity of tools we have now. I mean, we'll start with the traditional workhorse, the light microscope, or LM, and its sort of digital cousin, virtual microscopy.
But then we're going to get into the really high-res stuff.
Oh, yeah. We'll move pretty quickly to techniques that use electrons instead of light, like transmission and scanning electron microscopy, TEM and SEM. And then we get to explore the really cutting-edge tools.
Like Atomic Force Microscopy.
AFM, with its almost atomic resolution, and then the newest super resolution methods, which have, and this is amazing, literally found ways to break the fundamental physical limits of light itself.
It sounds like a complex journey, but understanding how these images are made is just it's completely essential.
It is, because if you're looking at a micrograph, you have to remember that every single image, it doesn't matter if it's a standard slide or some fancy electron micrograph, it's a two-dimensional slice.
And you have to mentally rebuild the 3D reality from that 2D image.
That's the skill. You have to learn to interpret that slice and mentally reconstruct how it fits back into the original three-dimensional biological structure.
Okay, let's unpack this whole process. We should start right at step one, getting the tissue specimen ready in the first place.
So the process really begins with the end goal in mind, which is creating that routinely prepared hematoxylin and eosin or H&E stained section.
That's the gold standard, right? The one that's studied pretty much everywhere.
It's a one. But you can't just take a piece of raw tissue and slice it. The first, and maybe the most crucial step, is fixation.
Fixation. So that's basically preserving the tissue's blueprint. What happens if you don't fix it, say, right after it's removed?
Well, it starts to degrade almost instantly. So the fixative, the chemical you use, has to do 4 critical things right away.
Okay, what are they?
1, it has to stop all ongoing cell metabolism. 2, it prevents enzymatic degradation, a process called autolysis, which is just self-digestion.
Yeah.
3, it kills any potential pathogens. And 4, maybe most practically, it hardens the tissue. It makes it rigid enough that you can actually slice it without it just falling apart.
And what's the go-to chemical for all of that?
That would be formalin. It's a buffered 37% aqueous solution of formaldehyde. It preserves the general structure incredibly well, mostly by chemically reacting with and cross-linking proteins.
It specifically targets the amino groups on proteins, right? Especially lysine residues.
Exactly. And that's a really critical insight because that specific cross-linking process is so valuable for things we might want to do later.
How so?
Well, formalin is effective because while it stabilizes the proteins, it doesn't really mess with their three-dimensional shape too much. And preserving that native structure is the bridge that lets us use modern, super-specific techniques later on.
Like immunocytochemistry.
Precisely. Because the antibodies you use in that technique need to recognize a very specific 3D shape, the epitope, to bind correctly. Formalin keeps that intact.
It makes sense. But if it's the gold standard, there have to be some limitations, right? Something it's not good at.
There is one major limitation, and every histologist has to know this. Formalin is, well, it's poor at fixing lipids.
Okay, so fats.
Right. Cell membranes, neutral lipids, they're just not well preserved. And then the next steps in the process make that problem even worse. Because when you introduce organic solvents, those lipids often just dissolve and are lost completely.
Which leads us right to that next phase, embedding and sectioning. So to prep the tissue for slicing, you have to replace all the water with something solid.
Exactly. That's the embedding process. So after fixation, the specimen gets washed to get rid of all the extra fixative, and then it's dehydrated.
How do you dehydrate it?
You run it through a whole series of alcohol solutions, each one with a higher concentration, all the way up to 100% alcohol to get every last bit of water out.
And then comes the clearing step. That's where those organic solvents you mentioned come in.
right? Specifically xylol or toluol. These solvents are key because they can mix with both the alcohol and the final embedding medium, which is paraffin wax.
So the xylol gets rid of the alcohol, and now the tissue is ready to be infiltrated.
Yep. It's placed into molten paraffin, the hot wax seeps into all the spaces within the tissue, and then you let it cool and harden. Now you have a solid, rigid block.
And that block can finally be sliced.
Right, on a machine called a microtome, which uses a very, very sharp steel knife. The sections that come off are amazingly thin, typically only 5 to 15 micrometers thick.
Just to put that in perspective, a micrometer is 1/1000th of a millimeter.
It's tiny. Then those thin sections get mounted permanently on glass slides using an adhesive like Canada Balsam or Permount.
But wait, we're not done. The tissue is still sitting in that colorless paraffin wax, and it's unstained. You can't see anything under a normal microscope yet.
Correct. So before you can stain it, you have to dissolve that paraffin wax out again using xylol or toluol, and then you have to rehydrate the tissue by passing it back down through the alcohol solutions into water.
And then we finally get to stain it with that famous H&E pair.
We start with hematoxylin, the H, which is applied in water. Then, to get the counter stain, eosin, the E, on there, you have to dehydrate the specimen again because eosin is dissolved in alcohol.
It sounds like a very precise chemical dance.
It is. It's a very specific sequence to make sure the stains stick where they're supposed to.
And the visual effects, which we can actually see in Figure 1.1, are incredibly informative.
They are. If you look at a section that's only stained with hematoxylin, you see this heavy blue or purple staining. And it's all concentrated in the nuclei and in the RNA-rich parts of the cytoplasm.
Those are the basophilic components, right? The base-loving ones.
Exactly. Now, in contrast, if you look at the slide with only eosin, the general cytoplasm and the matrix fibers outside the cells are all stained pink or red.
The acidophilic or acid-loving parts.
Right. And the nuclei are actually really hard to see with just eosin. So the real genius of the combined H&E stain is the incredible contrast it gives you. These dark blue nuclei against a pink background is why it's still the essential standard for seeing tissue architecture.
So we've established that the standard formalin prep with all those organic solvents gets rid of neutral lipids. When does that actually matter, and what do you do when you really need to see fat in a tissue?
It matters a lot. Anytime a pathologist is looking at, say, a fatty tumor or any tissue that's rich in triglycerides, to keep those neutral lipids, you have to skip the organic solvent step completely.
So how do you do that?
You have to prepare what are called frozen sections of the tissue and then use special fat-soluble dyes like oil redo.
And what if you need to see really fine membrane details, like for an electron microscope?
Then you need even more specialized fixatives. You have to use ones that contain heavy metals.
And the main one there is osmium tetroxide.
Exactly. Osmium tetroxide, or sometimes permanganate, is used because these heavy metals bind specifically to phospholipids. That stabilizes the membrane structures so they don't degrade or get washed away. It's the primary fixative for electron microscopy for exactly that reason.
Okay, now let's dive into that clinical correlation you mentioned, frozen sections. This isn't just about preserving lipids. This is a critical tool for making decisions during surgery.
Absolutely. Frozen sections are a necessity when a surgeon needs an immediate pathological diagnosis while the patient is still on the operating table.
So what's a common scenario for that?
It often happens when there's no preoperative diagnosis, but the most common use is to confirm that the margins of a surgical resection, meaning the very edges of the tissue that was removed, are clear of any cancer cells.
Getting clear margins.
That's the one. It's life or death information.
So how fast is this whole process, from the surgeon handing off the tissue to the pathologist calling back to the OR with a diagnosis?
It has to be extremely fast. We're talking as little as 10 minutes. The tissue is snap frozen, usually with cold isopentane at around -50 degrees Celsius, which turns it rock solid. Then it's sectioned inside a refrigerated cabinet called a cryostat, which is basically a microtome in a freezer. And then you do a very fast staining.
And while a quick H&E can be done, the sources show an example with a faster stain, methylene blue.
Right, figure F1.1.1 shows a great comparison. The frozen section stained with methylene blue. It might not have the beautiful, subtle color contrast of a full H&E prep.
But you can see what you need to see.
You can see the cellular architecture and any obvious pathology, like tumor cells invading. That rapid diagnosis is a critical, time-sensitive piece of communication between the pathologist and the surgeon.
You mentioned earlier that staining is all about chemistry, specifically electrical charge. Let's dig into that a little deeper, the actual mechanism of acidic versus basic dyes.
Okay, so you have to understand that these dyes are charged molecules. They create color by forming an electrostatic link with parts of the tissue. The fundamental concept is just opposites attract.
So let's start with the acidic dyes, like eosin. What are they attracted to?
Acidic dyes carry a net negative charge on their colored part. So naturally, they're attracted to and react with cationic groups in the tissue things that carry a net positive charge.
Which would be what?
Mostly the ionized amino groups of proteins. And this chemical reaction is called acidophilia, which just means acid-loving.
So eosin stains things pink because those structures are full of protein and have a positive charge.
Exactly. Acidophilia is what stains things like the unspecialized cytoplasm, filaments inside the cell, and fibers outside the cell, giving them that classic pink or red color.
And what about the opposite, the basic dyes?
Basic dyes carry a net positive charge, so they react with anionic components, the negatively charged parts of the tissue.
And in biology, that's mainly what?
It's dominated by two main chemical groups, the phosphate groups in nucleic acids, so DNA and RNA, and the sulfate groups you find in complex carbohydrates like glycosaminoglycans.
So things like cartilage?
Right, cartilage or the granules in mast cells. This reaction is called basophilia or base-loving.
So when we see that blue or purple staining from a basic dye, we know we're looking at something rich in genetic material like the nucleus, or something with a lot of synthetic machinery like the rough ER.
That's right. The rough ER, which was historically called ergastoplasm, is heavily stained because it's packed with ribosomal RNA. And that RNA provides a super high density of anionic phosphate groups for the basic dye to grab onto.
The sources also point out that you can manipulate this basophilic staining with pH. Why is that?
Because the ionization of those anionic groups depends on the pH. If you have a really high pH, like 10 or more, pretty much all the negative groups are ionized and ready to react. But if you lower the pH way down, say below 4, only the really strong acids, mostly the sulfate groups, stay ionized and can react.
So you can use pH to selectively target different macromolecules.
Exactly. It gives you another layer of control.
And what about hematoxylin, the H and H&E? Is that a true basic dye?
No, and this is the fascinating exception. Hematoxylin itself is not a strict basic dye. It actually needs an intermediate link called a mordant, which is usually a metallic ion like aluminum.
So the mordant is like a bridge.
It's a bridge. It links the dye to the tissue components, and that whole complex then acts like a basic dye, allowing it to bind to all those basophilic structures.
Why is that mordant so important to the H-ene process? Why not just use a simpler basic dye?
Because the final tissue mordant hematoxylin complex is much, much more stable than a simple electrostatic link.
Yeah.
It's so stable that it stays attached during all the watery and acidic washes that come before you add the eosin.
So a less stable dye would just wash away.
It might just dissociate and be lost. The mordant is key to the whole two-step process.
Okay, finally in this section, we have this phenomenon of metachromasia. This sounds like the dye is literally changing color.
It is. It's a chemical color shift.
Mm.
Metachromasia happens when certain basic dyes, like toluidine blue, encounter a tissue with a very, very high concentration of polyanions.
So a lot of negative charges packed really close together.
Think of the sulfate and phosphate groups in cartilage matrix, or inside the granules of mast cells. They're packed in there super tightly.
And instead of staining the expected blue color, what happens?
The color shifts dramatically to red or even purple. And this happens because that high density of negative charges forces the individual dye molecules so close together that they start to clump up, forming these dimeric and polymeric clusters.
And those clusters absorb light differently.
Exactly. They have totally different light absorption properties than the individual molecules, and that results in the visible color change. It's a really key diagnostic feature for identifying certain kinds of cells and matrices.
All right, moving into phase 2, we're shifting our focus a bit. We're going beyond that general architectural staining and into these highly specific chemical procedures, histochemistry and cytochemistry.
Right. These are the techniques that give us functional and compositional information about specific macromolecules inside the cell.
And the first major one is the periodic acid shift reaction, or PAS. This is our main tool for seeing carbohydrates, right?
It is. The PS reaction stains carbohydrates and carbohydrate-rich macromolecules a very distinctive magenta color.
So what are the key things it helps us see?
Its main applications are demonstrating glycogen, which, is the key energy store in cells like liver and muscle, as well as mucus, and some really important structural components like basement membranes and reticular fibers.
Let's walk through the chemistry that produces that specific magenta color.
It's quite elegant, really. It's a stepwise reaction. So the carbohydrate hexose rings have these adjacent carbons that have hydroxyl or amino groups. First, you add periodic acid.
Which acts as an oxidizing agent.
Right. It cleaves the bond between those adjacent carbons, and this cleavage creates highly reactive aldehyde groups.
Okay.
Second, these new aldehyde groups then react chemically with the Schiff reagent, which is a colorless solution of bleached basic fusin, to produce that visible deep magenta color.
And when we look at a micrograph, like the one of the kidney in figure 1.2, that magenta staining just pops. It really delineates the structures.
It does. Figure 1.2 clearly shows those PAS-positive basement membranes stained bright magenta, forming these really clear boundaries. You can see them surrounding the kidney tubules, the glomerular capillaries, and the Bowman's capsule.
And that's because those basement membranes are rich in proteoglycans and glycoproteins.
All of which have the carbohydrate components that the PAS reaction targets.
Then we have the Feulgen reaction, which is much more precise, targeting only DNA.
Right, the Feulgen reaction is specific to DNA because its target is deoxyribose. It uses a very mild hydrochloric acid hydrolysis.
So a gentle acid bath.
A very gentle one. It's just enough to cleave the purines from the deoxyribose of the DNA, which then opens up the sugar ring and forms, once again, aldehyde groups. And just like with PAS, those aldehydes react with the Schiff reagent to make the magenta product.
So what's the real significance of the Feulgen reaction? I mean, why not just use hematoxylin to stain the nucleus blue?
The significance is that it's quantifiable. The reaction with the Schiff reagent is stoichiometric,
Meaning
the amount of magenta color produced is directly and mathematically proportional to the precise amount of DNA in that nucleus. This makes it perfect for quantification. And crucially, RNA, which has ribose instead of deoxyribose, doesn't stain at all.
And how can a scientist be absolutely sure they're staining the right thing?
With enzyme digestion controls, it's a critical step. For example, if you see PA staining and you think it's glycogen, you could treat a control slide with an enzyme like diastase or amylase before you've seen it.
And if the staining disappears,
Then the enzyme digested the glycogen, and you've confirmed its identity. Similarly, you can use DNase to abolish Feulgen staining or RNase to get rid of the basophilic staining in the rough ER. These controls are essential for rigorous science.
that quantifiable nature of the Feulgen reaction leads directly into a key functional application, DNA quantification.
Yes, because it's stoichiometric, it lets us perform precise measurements of DNA, which is the whole point of Feulgen microspectrophotometry.
And the purpose of that is to analyze the ploidy of a cell population.
Correct. Ploidy is just the number of times the normal DNA content is multiplied. So a normal, non-dividing cell is diploid, or 2N. But cancer cells are often aneuploid, meaning they have these weird non-integral multiples of DNA, or sometimes they're tetraploid 4N.
And figuring that out is really important in oncology.
Hugely. The sources describe two main ways of doing this quantification.
What's the first one?
The first is static cytometry. This involves measuring the light absorption in a Feulgen-stained tissue section, like a tumor biopsy, using a microspectrophotometer that's hooked up to imaging software. It precisely measures how much light is absorbed to determine the DNA concentration compared to normal cells on the same slide.
And the second, faster method is flow cytometry.
Right. Flow cytometry is for cells that are suspended in a liquid. Instruments scan thousands of individual cells, one by one, very rapidly. And this technique usually measures fluorescent light coming off a specific dye that binds to DNA, rather than measuring absorption from the Feulgen stain.
So why does a pathologist care so much about the ploidy pattern? What's the big clinical insight here?
The big insight is that you're using a simple physical measurement, light absorption or fluorescence, to figure out how malignant a tumor is. Pathologists use these methods to analyze ploidy in all kinds of cancers: breast, colon, endometrial, kidney.
And how does that relate to a patient's prognosis?
Well, tumors that still have a mostly diploid pattern are generally considered well-differentiated. They tend to have a better prognosis.
And the opposite for aneuploid tumors.
Exactly. Tumors that show aneuploid or tetraploid patterns are usually more aggressive and are associated with a poorer prognosis. So this quantification gives crucial information that helps guide treatment plans.
Okay, let's shift from static molecules like DNA to dynamic processes. Enzyme histochemistry lets us see where specific enzyme activity is happening.
Right, and the core principle here is really important to get straight. You're not actually seeing the enzyme molecule itself. You're seeing the reaction product that the enzyme creates.
And to do that, the tissue has to be fixed very gently.
Very mildly, usually with an aldehyde. To preserve that fragile 3D structure, the enzyme needs to stay active.
And you use a capture reagent to trap the product right where it's made.
Precisely. You give the tissue the substrate for the enzyme, and you also provide a trapping agent, usually a dye or a heavy metal. This agent grabs the product and makes it precipitate immediately, right at the site of the enzyme.
The textbook gives a classic example of this from cell biology history, using acid phosphatase activity to identify the lysosome.
That's a fantastic example. In a lightly fixed tissue, you expose the section to a substrate that the lysosomal enzymes can break down. The reaction mix also contains lead ions.
And the lead ions are the trapping agent.
Exactly. As the enzyme works, the lead ions trap the product, forming this electron-dense lead phosphate precipitate. Seeing that precipitate inside these little vesicles confirmed the existence and the function of the lysosome.
And we can see this in an electron micrograph like figure 1.3a, which shows the localization of membrane ATPase.
Yes, in figure 1.3a, you can see these specific, dark, electron-dense areas that are located precisely along the plasma membrane of epithelial cells. That dark area shows where the ATPase enzyme is active.
And since we know that enzyme is the sodium pump,
we can confirm that this specific part of the cell is responsible for active transport and ion balance in that tissue. It connects structure, enzyme activity, and function perfectly.
Another really common method involves horseradish peroxidase, or HRP.
HRP is very popular. Its substrate, which is abbreviated DAB, produces this really striking brown, insoluble product when the enzyme oxidizes it.
And that brown precipitate is easy to see.
Very easy to see with a light microscope, and it also gives great contrast for electron microscopy. Figure 1.3b shows this beautifully. You can see kidney macrophages stained dark brown using the HRP-DAB method, which makes them stand out clearly against the light blue hematoxylin counterstain.
All right, we've moved from general chemistry to enzyme activity. And now we get to the ultimate level of molecular recognition, immunocytochemistry. This is all about the precise lock-and-key binding of an antibody to its specific target, the antigen.
This was the specificity revolution in histology. Antibodies are these large glycoproteins, immunoglobulins, that our immune system makes. We can purify them and chemically link or conjugate them to a detectable marker.
Usually a fluorescent dye, a fluorochrome.
Right. Fluorescein is a really common one because it absorbs UV light and then emits a bright green light that's easy to see.
The sources highlight two main types of antibodies that are used in the lab.
First, you have polyclonal antibodies. These are basically a mixed bag of antibodies produced by many different B lymphocyte clones in an animal that's been immunized. Because it's a mix, they recognize multiple different regions or epitopes on the target antigen.
And then came the real game changer. Monoclonal antibodies.
Monoclonals were revolutionary because they're produced by a single immortalized cell line called a hybridoma. This means every single antibody in the batch recognizes only one specific epitope on the antigen.
Which gives you incredible specificity.
Unparalleled specificity and standardization. It's essential for both research and diagnostic tests.
Speaking of diagnostics, monoclonals have huge applications in medicine. They're not just research tools.
Oh, not at all. They're absolutely central to modern medicine. They're used all the time for diagnosis, detecting tiny tumor metastases, differentiating between very similar tumor types, identifying infectious diseases.
And they can be used therapeutically, too.
Yes. You can engineer them to deliver a payload. For example, you can link them to toxins or chemotherapy drugs or radioisotopes to deliver those agents directly to tumor cells, which maximize the effect on the cancer while sparing healthy tissues.
When we apply these antibodies to tissue, we have two main ways to see them, direct and indirect.
Right. Direct immunofluorescence, which you can see in figure 1.5a, is the simplest method. The fluorescent dye is attached directly to the primary antibody that binds the antigen. It's just one step, very quick.
But the drawback is a weak signal.
A very low signal intensity because you only get one fluorescent molecule for every one antigen molecule.
And that's why the indirect immunofluorescence method, the sandwich technique, is so much more common.
The indirect method is vastly superior because it gives you this dramatic signal amplification. You use an unlabeled primary antibody first that binds to your antigen. Then you add a second fluorescently labeled secondary antibody.
And that secondary antibody is designed to bind to the primary one.
Exactly. It's an anti-antibody. So if your primary was made in a mouse, you'd use a goat anti-mouse secondary. And since multiple secondary antibodies can bind to a single primary antibody, the fluorescent signal gets amplified considerably. You get much brighter, clearer images.
This amplification also lets you visualize multiple different things in the same section at the same time, like in Figure 1.6.
Figure 1.6 is a beautiful demonstration of that. You're looking at HeLa cells, where the microtubules are labeled in bright green and certain nuclear proteins are labeled in magenta, all on top of the blue DAPI-stained DNA. This kind of multi-channel image lets researchers study how different proteins are localized relative to each other with incredible confidence in detail.
We are now moving into what I think is the most exciting territory: molecular recognition that focuses on nucleic acids, and methods that are designed to physically break the limits of what we can see. We're starting with the power of sequence matching, hybridization.
At its core, hybridization is just the physical binding of two single-stranded nucleic acids, the sequence you're interested in, and a lab-created complementary sequence that we call in situ hybridization.
In situ meaning in its original place. So in situ hybridization lets us find specific DNA or mRNA sequences right inside the cells or tissues where they live. We label our probes with different markers, maybe radioactive isotopes, or more commonly now, non-radioactive labels like digoxygenin or biotin. So you can track gene expression or gene location.
Exactly. And techniques like PCR and RT-PCR are crucial here for getting a strong enough signal.
Right, because you might be looking for just a tiny amount of your target sequence.
Yes, even if there's only a single mRNA molecule, you can use PCR or RT-PCR to amplify that target, making it much easier for the probes to find and for us to detect.
The most common modern version of this, fluorescence in situ hybridization, or FESH, has totally revolutionized genetic testing.
It has, because it allows for multiple colors at the same time. FSH combines fluorescent dyes with these nucleotide probes, so you can light up 2, 3, or even more different genetic sequences simultaneously using different colors.
The classic example of this, which we can see in Figure 1.7, is in prenatal testing for chromosomal abnormalities.
Figure 1.7 gives you this immediate visual diagnosis. On the right, you see a normal nucleus. It's got 2 green signals for chromosome 13 and 2 orange signals for chromosome 21 and normal set, but the nucleus on the left.
It has 3 orange signals.
3 orange signals. That's trisomy 21, or Down syndrome. FISH gives you a fast, unambiguous way to count chromosomes. And beyond prenatal testing, it's used in cervical cancer screening to look for viral DNA, and even to assess radiation damage in astronauts by checking for chromosome breaks and translocations.
Now let's talk about autoradiography. It's an older technique, but it's still fundamentally powerful for tracing dynamic cellular processes, like synthesis.
It's an indispensable method for tracking where and when macromolecules are being made. The whole mechanism relies on introducing a small molecular precursor, say radioactive thymidine for DNA synthesis, or radioactive leucine for protein synthesis.
So you're tagging the building blocks.
You're tagging the building blocks. The living cells take up this radioactive precursor and build it into new macromolecules. Then you fix and section the tissue.
And how does that radioactive tag become a visible image?
This is the clever part. You dip the slide in a melted photographic emulsion, basically coating it in a layer of undeveloped film. Then you wait. Over days or weeks, the radioactive emissions from those tagged atoms expose the silver halide crystals in the emulsion.
So it's like the cells are taking their own picture.
In a way, yes. After you develop it, the exposed silver halide crystals turn into metallic silver, which show up as these little dark grains right on top of where the radioactive material was located.
Figure 1.8a shows a really clear example of this with light microscopy.
Yeah, that's a lymph node section where they were tracking cell division using radioactive thymidine. You can clearly see clusters of these little black silver grains specifically over the nuclei of the cells that were actively making DNA to get ready to divide.
So you can literally see which cells are proliferating.
It's a direct measure of cell proliferation. And the same principle can be scaled up to much higher resolution with EM autoradiography.
So using an electron microscope.
Right. Figure 1.8b is an electron micrograph that lets us see the precise subcellular location of these tagged molecules. The silver grains are much smaller, and you can see they're concentrated right over the apical invaginations and endosomal tubules in an intestinal cell. It's crucial for tracing the exact intracellular pathways of specific molecules.
This brings us to a technique that just feels like science fiction: expansion microscopy, or XM. The ultimate physical limitation of light microscopy has always been that diffraction barrier, that 0.2 micrometer limit.
Right.
And XM just finds a way around it by physically stretching the specimen itself.
It is the definition of a disruptive technology. Instead of trying to make a better microscope, XM makes a better sample. It physically expands the tissue, pulling those previously crowded structures so far apart that they're no longer limited by diffraction, and you can resolve them with a normal light microscope.
Let's break down the steps, which are outlined in Figure 1.9. It uses those super absorbent materials, hydrogels.
That's right. The tissue gets infiltrated with monomers of a hydrogel, a swellable polymer, like sodium polyacrylate. There are 4 major steps before you get to the actual expansion.
What are those prep steps?
First is anchoring. The proteins you're interested in, which are usually fluorescently labeled, get chemically linked or anchored to the monomer molecules that are going to form this polymer web. Second is gelation, where the monomers polymerize into a dense, cross-linked hydrophilic 3D matrix that is now physically attached to everything inside the cell.
And then you have to get rid of the original cell structure.
Yes. The third step is mechanical homogenization. You break open the cells and use enzymes, proteases, to digest all the structural molecules. This is key because it ensures that the only thing holding all the labeled components in their relative positions is that artificial hydrogel matrix.
And then comes the magic.
Then comes the expansion. You just add pure water. The hydrogel is super hydrophilic, so it sucks up the water and swells, causing this isotropic expansion. It pulls apart equally in all 3 directions and increases the volume by more than 100-fold.
And what does that buy you in terms of actual resolution?
A huge gain. You can routinely get a linear expansion of up to 4.5 times, which pushes the light microscope's resolution down into the 60 to 70 nanometer range.
Which is incredible.
It is. And if you repeat the process with what's called iterative expansion microscopy, or IEXM, you can get up to 20 times linear expansion, giving you resolutions around 25 nanometers, which truly starts to rival a traditional electron microscope.
The sources mention the implication for diagnostics with something called expansion pathology, or XPath. This seems massive.
It is completely transformative. XPath is about applying these protocols to routine clinical H&E slides. Just look at the comparison in Figure 1.10. Panel A is a normal H&E, and B is an immunofluorescent image of mammary gland tissue. Both are at 40x magnification. The nuclei are all crowded together.
But then you look at panel C.
Panel C is the adjacent section processed with XXM, imaged at the exact same magnification. And the improvement is just dramatic. Structures that were crowded, like the nuclei, are now clearly distinguished. This allows for optical diagnosis at a resolution that you used to need an EM for.
So it could potentially replace the need for an expensive EM in some clinical settings?
That's the potential. It could fundamentally change how we do diagnostics.
We've explored some of the most cutting-edge methods, but the foundation of all of this is still the light microscope. So we should probably step back and make sure we really understand the fundamental optical concepts that govern all of these techniques.
Absolutely. When we talk about microscopes, the most critical concept isn't magnification. It's resolving power, or resolution.
And resolution is what, exactly?
It's the smallest distance that 2 objects can be separated and still be seen as 2 distinct objects. The resolving power of the naked human eye is about 0.2 millimeters.
And how much better is a standard bright field microscope?
It's a 1,000-fold improvement. The theoretical limit of a bright field microscope is about 0.2 micrometers. And it's important to remember, while the eyepiece, the ocular lens, magnifies the image, it does not increase the resolution. Resolution is fundamentally determined by the objective lens and the wavelength of light you're using.
And how you align the illumination.
And proper illumination alignment, yes.
You stressed earlier that every histologist has to overcome this 3D challenge.
This is often the steepest learning curve for students. A histologic section is just a 2D slice of a 3D organ, and the way structures look can vary drastically depending on where you cut. Figure 1.11 uses the great analogy of slicing an orange.
Right, a slice through the middle looks like a perfect circle.
But a tangential slice near the edge might look like a crescent or just an incomplete shape.
And this translates directly to structures we study, like the renal corpuscle in the kidney.
Precisely. Depending on whether your 5-micrometer slice goes through the exact center of that spherical corpuscle, it might look like a full ball of capillaries. But if you just graze the edge, it might look like a small ring of cells or a dense half-moon shape. You have to look at many, many 2D sections to mentally piece together the full 3D structure.
And finally, we have to mention artifacts, those things that confuse every beginner.
Artifacts are just defects. They're caused by errors in the preparation. Maybe the fixation time was wrong, or the reagents were contaminated, or there was physical damage from the knife, or poor mounting. A good histologist has to learn to recognize these flaws, like shrunken cells or folded tissue, so they don't mistake them for real biological structures.
So if resolution is king, then optimizing the microscope is the only way to get there. And the sources detail this indispensable process called Kohler illumination.
Kohler illumination is the standard protocol for getting the best performance out of your microscope. It ensures that the illumination and the observation light paths are perfectly aligned and centered. Without it, even the best, most expensive objectives will give you suboptimal resolution and contrast.
It's a 5-step process, and it starts with focusing on the specimen and then playing with the diaphragms.
Right. After you focus, the critical step is to adjust the field diaphragm. You close it all the way down, and then you focus and center the condenser until the outline of that diaphragm is sharp and perfectly centered in your view.
And then you open it back up.
You open it just enough to fill your field of view. The point is to make sure you're only illuminating the area you're actually looking at, which prevents stray light from washing out the contrast.
And the final critical step involves adjusting the condenser diaphragm, which is where you manage that trade-off between resolution and contrast.
This is where you strike the balance. You want to open the condenser diaphragm, the illuminating aperture, until it covers about 2/3 of the objective aperture. If you open it too wide, your contrast plummets. If you close it too much, you get better contrast, but you start to introduce ugly artifacts like diffraction rings. 2/3 is the sweet spot.
The sources show the formula for calculating the resolved distance, and it really proves why the condenser is just as important as the objective.
It does. The formula shows that the numerical apertures, or NA, of both the objective and the condenser contribute equally to achieving the maximum possible resolution. You can have the best objective in the world, but if your condenser isn't properly aligned and set, you're not getting its full power.
So why does closing that condenser aperture a bit actually enhance the contrast?
It has to do with the light paths. Closing the aperture brings the intensity relationship between the light that's diffracted by the specimen and the non-diffracted background light closer to a 1-to-1 ratio. It allows for optimal interference, which is what maximizes the visible contrast in the final image.
Okay, beyond the standard bright field, there's a whole catalog of specialized optical systems designed to see things that are otherwise invisible.
We can start with phase contrast microscopy. This is a technique for unstained samples. It exploits tiny differences in the refractive index, the density of different parts of the cell. It converts those invisible density differences into visible contrast.
So it's perfect for looking at living cells.
Exactly. It's extremely useful for observing unstained living cells in culture.
Then there's dark field microscopy.
Dark field uses a special stop in the condenser to block the direct light. It illuminates the specimen from the side, obliquely, so that only the light that's scattered or diffracted by structures actually makes it to the objective.
So the specimen looks bright against a black background.
Right. And this makes it perfect for detecting really small, fine particles that would be invisible in bright field things like spirochetes, tiny crystals, or those silver grains we talked about in autoradiographs.
Fluorescence microscopy is foundational for almost everything we've talked about in the last 2 phases.
It is the absolute workhorse of molecular imaging. It uses a high-energy UV light source to excite fluorophores molecules that absorb that UV light and then immediately emit visible light at a longer, less energetic wavelength. And those fluorophores are usually attached to antibodies. Far more commonly, yes, they're tagged onto antibodies for immunocytochemistry.
Now we're getting into the advanced tools that let us optically dissect tissues. Light sheet fluorescence microscopy, LSFM, is one of the newest.
LSFM is an imaging method that uses a very thin, flat laser beam, the light sheet, to optically section a transparent, fluorescently labeled specimen. And the key is that the emitted light is collected perpendicularly to that light sheet.
So you're only illuminating the single plane you're looking at.
Exactly, which minimizes the damage to out-of-focus areas and dramatically reduces phototoxicity. This lets you do long-term imaging of live, large specimens. The result, which you see in Figure 1.12, is a remarkable 3D reconstruction of galanin-positive cells in a rat spinal cord, all built from stacks of these clear, optically sectioned images.
The other key optical sectioning tool is the confocal scanning microscope. What's the fundamental innovation that makes it confocal?
The central innovation is the detector aperture, or pinhole. It's placed in a position that is conjugate or confocal with the focal point of the objective lens.
And why is that pinhole so powerful?
As you can see in Figure 1.13, that pinhole acts as a spatial filter. It physically blocks all the light that's scattered or emitted from structures above or below the focal plane. All that out-of-focus light gets rejected. Only the light coming precisely from the focal plane can pass through and reach the detector.
And that gives you incredible clarity.
Exceptional clarity. The image isn't viewed directly, though. As you can see in Figure 1.14, the confocal uses a laser and scanning mirrors to excite the fluorophores spot by spot across the specimen. A computer then takes the signal detected through that pinhole and reconstructs the image, sort of like a CE scan.
And we also have the polarizing microscope for those really highly ordered crystalline structures.
Right. This is a light microscope with two polarized filters. It's designed to detect birefringence, which is the ability of highly ordered molecular arrays to rotate the plane of polarized light. It's invaluable for identifying things like the contractile filaments in striated muscle or collagen fibers.
And finally, we have to talk about pushing past that zero point 0.2 micrometer limit, entirely super resolution microscopy.
This is a whole group of methods that achieve resolutions down to 10 to 100 nanometers. They do it using computational and physical tricks to isolate single photons or manipulate the light spot size.
Like STED microscopy.
STED, or stimulated emission depletion, uses two lasers. One excites a fluorescent spot, and a second donut-shaped depletion laser forces the excited molecules at the edge of the spot back to the ground state. This effectively shrinks the size of the fluorescent spot far below the diffraction limit, getting you down to 30 to 80 nanometer resolution.
And what about the single molecule methods like PAL and STORM?
PALM and STORM rely on photoactivatable fluorescent molecules. The key insight is, if you only activate a tiny statistically resolvable handful of molecules at any one time, you can locate the center of each one with extreme precision. You cycle this process, activate, locate, deactivate thousands of times, and a computer compiles all that data to build an image with resolutions approaching 10 to 20 nanometers.
Okay, now we make the final jump in resolution, moving to electron microscopy, which offers another 1,000-fold increase in resolution over light microscopy by using beams of electrons instead of light.
The Transmission Electron Microscope, or TEM, uses a beam of electrons focused by electromagnetic lenses. The electron beam has this incredibly tiny wavelength, about 1/2000th that of light, which gives it a theoretical resolution of 0.05 nanometers.
The prep for TEM must be way more rigorous than for light microscopy, given that level of resolution.
Oh, it requires supreme fixation and embedding. You start with glutaraldehyde for the proteins, but then you immediately follow that with osmium tetroxide. The osmium is crucial because it binds to lipids, stabilizing membranes, and it imparts high electron density, which you need for contrast.
And the samples have to be tiny.
Exceptionally small, maybe 1 millimeter cubed, and embedded in a hard epoxy resin.
And the sectioning must be incredibly difficult.
It is demanding. The sections have to be ultra-thin, only 50 to 150 nanometers, because electrons just can't penetrate very far. You have to use diamond knives, not steel, and the sections are mounted on little copper mesh grids.
And staining is done with heavy metals.
Right, solutions of things like uranyl nitrate and lead citrate, which scatter the electrons and increase the contrast.
The sources also mention freeze fracture, a special TEM technique for looking at membranes.
Freeze fracture is designed to reveal the internal architecture of a membrane. You rapidly freeze the tissue and then fracture it in a vacuum. The fracture plane naturally follows the path of least resistance, which is right through the hydrophobic core of the plasma membrane, splitting it into its two internal leaflets.
And what do we call those two exposed faces?
They're called the E face for extracellular and the P face for protoplasm. You then make a platinum and carbon replica of that exposed surface, dissolve away the actual tissue, and look at the replica in the TEM. It's the only way to really see the integral membrane proteins embedded within the membrane structure.
Okay, then there's the Scanning Electron Microscope, or SEM, which works on a completely different principle and gives us that famous 3D surface view.
That's right. Instead of the electrons passing through the specimen, the electron beam is scanned across its surface. Detectors then collect the electrons that are reflected back or knocked out of the surface to generate a high-resolution, three-dimensional topographical image.
The resolution is a bit lower than TEM, but the 3D rendering is incredible.
It is. The preparation involved fixing and dehydrating the sample and then coating it with an electrically conductive layer, usually a gold carbon film, to prevent charge from building up.
And now we're seeing the rise of three-dimensional electron microscopy, or 3DEM. This is for when you need to see how entire cells and organelles are connected.
Yes, 3DEM is essential for capturing biological reality. The simplest, though most laborious approach is serial section TEM, where you physically cut, collect, and image hundreds of consecutive ultra-thin sections and then digitally reconstruct them.
And that labor is why the newer automated methods built into the SEM are so important, like Serial Block-Face SEM.
SBF SM is a major advance. It puts a precise ultramicrotome right inside the SEM vacuum chamber. The whole cycle is automated. The microtome shaves off a super thin slice, say 20 nanometers, and immediately the electron beam images the newly exposed block face before the next cut. This process repeats thousands of times, generating these massive, perfectly aligned stacks of images automatically.
Figure 1.1 sec shows the final critical step in this, image segmentation.
Right. Segmentation is the post-processing step, where you assign a label to every pixel associated with a specific structure you're interested in. In the figure, the nucleus is segmented and colored blue, the plasma membrane green. This lets researchers isolate and model those structures in 3D. It can be a tedious manual process, but it's essential for making sense of the data.
Now, for a really remarkable technique, Atomic Force Microscopy, or AFM, it's not optical, it's not electron-based, and yet it gets molecular or even atomic resolution, and it doesn't need a vacuum.
The AFM works literally like a fingertip tracing a surface. It uses an ultra-sharp probe on a highly flexible cantilever to scan the specimen, reading the tiny atomic forces on the surface.
And how is that physical movement turned into a picture?
A laser beam is reflected off the top of the cantilever onto a photodiode detector. When the tip hits a bump, the cantilever deflects, and the photodiode precisely measures that tiny deflection. A computer records the adjustment needed to maintain a constant force and translates that into a high-resolution topographic image.
And the resolution is incredible.
We're talking molecular and atomic resolution, down to 50 picometers. And the major biological advantage is that you can image specimens in their native state. You can look at living cells submerged in water.
Figure 1.1 each shows an amazing visualization of this.
It does. It shows a single DNA molecule lying on a mica surface. You can see the strand itself, and where binding proteins are attached, they create these little thickenings or bumps. AFM provides a way to visualize macromolecular interactions in real time in a natural physiological environment.
Okay, let's wrap up with virtual microscopy, or VM, which is transforming how histology is taught and practiced.
VM just integrates the conventional light microscope with high-resolution digital tech. Instead of looking through an eyepiece, entire glass slides are scanned at high magnification to create huge 2D digital files, the virtual slides.
For a learner, this is a massive leap in accessibility.
The advantages are immense. You get remote viewing on any device, seamless zooming from low to high power without changing lenses, an orientation map so you never get lost, and you can make your own annotations and drawings right on the digital slide.
And clinically, this drives the practice of telepathology.
That's right. Virtual slides can be shared instantly anywhere in the world, allowing pathologists to render diagnoses from a distance. It's quickly becoming the new standard for tissue examination. So what we've covered today is really a comprehensive methodological evolution. We started with the basic necessity of fixing tissue with formalin, relying on simple acid-Dece chemistry to get the contrast of an H&E stain.
And from there, we moved into highly specific localization tools like PAS and Feulgen, and then the immense specificity of the antigen-antibody reaction in immunocytochemistry.
Exactly. And we completed our journey in the 21st century, moving from molecular localization with FESH to the computational imaging revolution, the tools that deal with the physical barriers of light, like confocal and super resolution.
And then to the ultimate resolution of electrons and physical probing with TEM, SEM, and AFM. And we tracked the convergence of these fields with techniques like 3DEM and the, I think, ingenious simplicity of expansion microscopy.
The convergence of optics, chemistry, and computation is the major theme. It's what allows us to ask and answer biological questions with a resolution that was just unimaginable even a decade ago.
So here's where it gets really interesting, and this is the thought I want to leave you with. Consider how the increasing resolution and 3D reconstruction capabilities, especially with techniques like expansion pathology applied to routine, inexpensive H&E slides, are rapidly blurring the lines between structural histology and molecular biology. If we can achieve 25 to 70 nanometer resolution using relatively inexpensive light microscopes, which are in almost every hospital, what is the ultimate future role of those traditional vacuum-dependent TEM labs in clinical diagnostics?
That's a great question.
This ability to see molecular detail through conventional optics fundamentally changes the way we study and diagnose disease.
It absolutely does. That's an excellent question for all of us to consider as these technologies continue to mature.
Thank you for joining us for this deep dive into the foundational and cutting-edge methods of microscopic anatomy. We hope you now feel fully equipped to understand not just the biological structures you see, but how those spectacular images are truly made.